So as I said, Jennifer did really a good, very nice talk about inversions before me, but I'm going to explore a little bit more on this topic. And we have been very interested in our lab in studying inversions. I will explain the motivation a little later in the slide. But one of the things we need to think about is that inversions were cytogenetically defined some decades ago as copy number neutral events that change the direction of the strand when compared to another genome. In the case we are comparing to the reference. A very important aspect of the inversions is that they are formed by two breakpoint junctions. And that is key for detection of inversions because we really need to detect both of these breakpoint junctions to make sure we have an inversion and to confirm the presence of such an event. And that can be a challenge sometimes. So why we want to study inversions? So first, inversions in general, although we know them for a very long time, they are actually not very well characterized in the genome, and we don't really, many times we just ignore them from the short-read data because there are a lot of false positives and false negatives in the bulk of the data. So a recent paper, very nice group paper about inversions and other structure variants by Charles Lee group, shown that on average, a person has around 156 inversions, each one of us. So this is, of course, potentially non-pathogenic inversions, but we need to think about carriers of inversions that disrupt genes, that when homozygous can cause disease, and also the fact that de novo inversions can be formed in once every 10,000 pregnancies, and the fact that de novo inversions can be associated with congenital anomalies in 9.6% of the patients that are referred to a diagnostic lab. That's an important number, and I think is very relevant, and we need to remember these numbers were derived from cytogenetics, so we really don't know yet what is this percentage using next-generation sequencing. And as I said, although we know that they are there and they are important and relevant for disease, we still don't have a good picture of inversions and a very good way to detect them, and also it's challenging to confirm them using a different technology, an orthogonal technology. Very briefly, I'm going to mention the two main mechanisms that are involved in the formation of inversions. One of them is non-allelic homologous recombination, which Jennifer explored a lot on her talk, and non-homologous joining. In non-allelic homologous recombination, we do have a formation of inversions by ectopic recombination between two inverted repeats. So the breakpoint junctions will be inside the repeats. This is a very important mechanism for formation of inversions because we know that the human genome is formed by around 50% of it. It contains repeats. The local repeats that Jennifer mentioned, they are for more or less 5% of the genome. Non-homologous joining can form also inversions by double-strand breaks in cis in the same strand. And then the junctions will be formed by repair and ligation. And usually these junctions are very straightforward to find because they are usually blunt or they have like small insertions or deletion at the junction. So these ones are more easily to be detected by next generation sequence than those here. And in fact, previous work shown that non-homologous joining can contribute to about 30 to 44% of the inversion formation, and non-allelic homologous recombination can contribute to about 55 to 70% of the inversion formation. We have a new work that is ongoing, and we are about to submit in collaboration with the Karolinska group led by Anna Lindström, that is showing that, in fact, inversions formed by non-homologous and joining are easily detected by short reads, but not inversions formed by non-allelic homologous recombination. And that's why we can not really easily detect inversions formed by this mechanism. So to be very, to summarize the observations that we are blind to this inversion. That's exactly what Jennifer just talked on before me. And why? Because the inversions formed by non-allelic homologous recombination, they map the breakpoint junctions inside these repeats, and they are not copy number, they usually are not copy number, they don't have copy number event along with. So ReadDep is not going to detect them. Also, because the repeats are usually can be as big as small as 300 base pairs in an ALU event, for example, or as big as 300 kb or 500 kb. The next generation sequence, if you use like short reads, is not going to be detecting these inversions by this code and read pair or split reads. So we cannot, basically we are not detecting them at all in our work if you are just using short reads. So as has been talking already today, the optical mapping does not does sequencing of nucleotide, but it does it does label specific sequence motifs in single DNA molecules. The technique we are using now is the direct label instant technology, the new one, DLS. And using this protocol, we can actually map the motifs in the genome, and this is the reference in green. I will show the same color in the next slides. And our sample, and can make a sample alignment using genome maps that are generated with this technology. The key and very important thing about this technology, and that's why we decided to try to detect the inversions by non-allelic homologous recombination, is that it does produce a very large DNA molecule of about 100 and 200 kb, and that we can use this to detect at least a great part of the structure variants that fall within this category. That means then we can cross some of the low-cop repeats and some of the repeats using it. So the optical mapping preserves the architecture of the region, so we can use, we can detect both breakpoint junctions that will be in cis by analyzing the molecules and the raw molecules that the technology produces. So the hypothesis of our study is that long molecules will enable detection of copy number neutral inversions mediated by repeats, and long molecules may increase the precision of the inversion calls since longer molecules have higher probability of bearing both junction 1 and junction 2 of an inversion. So to do that and to test our both of our hypotheses, we decided to start using a sample that was very well studied in by previous group, and this is a Coriell sample. NEA1510 is a female sample that was studied by Tuzun in 2005 using pair-end phosmids, was studied by Kidd et al. In 2008 using the same phosmids, or some of them, they actually sequenced the whole phosmid, and also by Carvalho using solid sequencing. And then so we have a very good benchmark to try to test optical mapping against this. We also decided to develop a tool to identify inverted repeats more quickly in the human genome, because we all use the segmental duplication track for doing that. However, the segmental duplication track has some limitations, and one of them is that to really learn that segmental dup is an inversion, we need to kind of explore the data of the segmental duplication in deep. But we wanted to quickly find these inverted repeat pairs and use this data to construct our bed file in Access in the Bionano algorithm. So another thing we wanted is to unmask. We want to unmask the repeats that are repetitive elements because they are about 50% of the genome. So the segmental duplication tracks, they mask those repeats, so we don't see them. For example, lines are around 6 kb in size, so they are and we know that they cause deletions and duplications by ectopic recombination. So of course they are, not of course, but we predict they are potentially causing inversions as well. But we are not detecting them like promptly. So we developed this tool that is going to be published soon in Human Mutation. And this tool actually is does a pairwise alignment. It imposes no restriction in size on the detect repeat fragment and allows 95 to 100 identity levels between the pairs. And more important, it does not use filtering of the repetitive elements. So in total, we identified 59,000 of repeat pairs. They can be direct or inverted. And so half of them are inverted repeats that are larger than 700 base pair in the whole genome. So here's just an example. This region here is close to MACP2. MACP2 is a gene very important that causes MACP2 duplication syndrome. We work with this disease a lot in the lab. MACP2 is around here, but these inverted repeats shown by the segmental duplication track in orange are very important for the formation of a lot of the rearrangements in this region that includes triplication that has inversions as well. And with the tool that we developed, we can easily and quickly see that these are the same repeat pairs. So that's just like for convenience. But this one is a very important example. Close to PLP1, which is another locus that also have rearrangements involving inverted repeats and cause a disease called Pelizaeus-Merzbacher disease, we found a lot of lines of around 6 kb, and they are inverted repeats to each other, and the distance is around 150 kb. So they can easily undergo ectopic recombination and invert this region. So with this, we start building our pipeline to analyze the inversions on the sample NA1510. Here is just how we are doing the analysis, but I think everybody already probably went over this. And we can easily see the inversion, the inverted orientation of the sequence motifs compared to from the reference with our sample. And we use the raw molecules to make sure we can see in the same molecule the breakpoints in cis. Here is how we are doing our analysis using Access. So we are adding, so this is the optical mapping data showing the breakpoint junctions of the inversions. Here is our track that with the tool we are using. This region actually is very well mapped by the segmental duplication track. You can see that the inverted repeats that we revealed are mapped very well with the segdup track. Of course, it doesn't map with the whole thing because probably those here are potentially direct repeats or maybe have similarity that's lower than what we put as a threshold, that's 95%. But still, we could see the breakpoint junctions very well and mapping to the inverted repeat pairs that we mapped. So here we also enter with the bed files we got from the GV, from the other papers, from the other work, Estuzum, Kidd, and Corbell. And an important thing that I want to point out here is that the breakpoint junctions that those technologies that they used map, they are not precisely mapping to the correct place, so which would be the inverted repeats. So we can see a discrepancy between what the inversion, despite all of the technology found an inversion, the junctions are not precisely mapped. And this is expected because this technology that they use here, they cannot really cross the junctions. So phosmids is like 45 kb, so the size of the repeats here is about 200 kb. So it's not possible to cross, so that they probably only found one of the junctions. So here is a summary of the data we observed for the Coriell sample. We use DLS technology. We detect about 46 inversions. The smallest size was 1.5 kb. The largest size, 3.8 megabase. And we observed that 1/4 of those inversions actually are mapping within the inverted repeats that we detected. And here is a comparative table showing that we got about the same order of magnitude of inversions compared to the other groups for the same sample. And it's very interesting that, as I mentioned here, the involvement of inverted repeats were very strikingly higher than the other groups. And I think it's especially interesting that Corbell, that used solid, got 6% only, kind of supporting our observation that we need long reads to be able to get the inversions that are mediated by repeats.
Quick question about that. So the largest and the smallest, were they the same throughout all four of those papers, or the 3 papers in your work?
The largest and the smallest size? No.
Yeah, the sizes are different, but are they the same regions?
Yeah, that's what I'm going to show you. You mean the overlap between?
Yes.
Yes, that's what you asked. So here we are just comparing optical mapping with those because KID and 2Zoom are very much almost the same. They use the same. KID actually select the sum of the FOSM that 2Zoom published, so they should be very similar. But Corbell is different. So we see that optical mapping is overlapping in a good portion with KID and 2Zoom, and not very much with Corbell, because Corbell is detecting more inversions that are probably formed by normals and joining. We still have to explore this data further. And KID and 2Zoom are using phosmid, so they are also longer inserts if we compare to the Corbell. That the size of the inserts in Corbell is like 3 kb compared to 45 in here and 150 in optical mapping.
I assume you have remapped all of that to the latest genome build, but obviously those are relatively old studies.
I know.
So how much of that
Yes.
is blame on the relatively
Yes.
reference genome, especially in those regions? You could imagine that there is differences.
Yes. So yes, 2Zoom was mapped to ages 17. We had to make a lift. So yes, I don't know the answer for this question. It's possible that some of this is explained by the mapping, but you mean the discordance between the breakpoints, right? Yes, we need to explore this better, for sure. And here I already talk and show about the difference in the size and in the difference in the breakpoint junctions. Here I am actually showing the size that this specific inversion that was detected by 3 out of the 4 technologies Corbell didn't detect. And we can see that the junction of optical mapping maps in the inverted repeats that flanking it very nicely. 2Zoom detect the same inversions, of course, but and KID, they both detect the inversion, but with a different breakpoint junction. So using this, we can precisely compare the original inversion with the location, and has like 100 kb of difference. Here is an example of an inversion that was detected by optical mapping, but not by the other technologies. And we also have inverted repeats flanking it, and we can detect and see the molecules that support the presence of the 2 breakpoints in cis. We also could see some phasing that, of course, we need to confirm, but we could see some phasing using optical mapping. However, we also observed that there are some limitations with the optical mapping data, and one of them was kind of a little bit frustrating for us because is that this region is close to MACP2, in exactly the one we were interested in, inversions we are interested in mapping and in detecting our patients because we work a lot with these samples. And as you can see, we know that 2Zoom and KID et al. Reported these inversions. We know that this region between these 2 inverted repeats are very important for our patients. So, and we already confirmed the presence of this inversion using also Southern blot. However, optical mapping, the DLS, there is no sequence motifs for the enzyme here, for the LA enzyme here, so we could not detect the inversion on these regions. So there are regions in the genome that do not have this motif, so we know there's be some limitations for this work. We also got some result that we don't know are false positives or false negatives. And in this case, all the three groups detected an inversion here. Because they detect different size, we don't know exactly if they are forming one junction, one inversion, like it all seems to be saying that there is just one inversion here, although Tuzum and Carvalho have different size. But we couldn't confirm with optical mapping. So we still have work to do and confirm and validate this inversion. So what we are planning, we are actually doing high-C on this sample. And we'll do some PCR because some of the inversions are not in repeat, so we can do PCR to check. And we may use some ddPCR. There is a new paper showing that ddPCR can be also used for inversions that have a distance of 100 kb or 200 kb or something. We'll see. So as conclusions, optical mapping provides a complementary tool to characterize previously challenged structure variants, in this case, copy number neutral inversions. The location size and genomic content of the previously documented inversions can be further refined using long reads and analyzing in-cis, long reads that can provide in-cis information. And of course, we need to still validate this data to be able to have a better idea about the numbers and the precision. So with this, I want to thank the group we are working with, the PhD student, Christopher Grunchowski, Claudia Gonzaga, who did the inverted repeat tool that I presented, Jesper and Anna from the Karolinska Institute that are working with us in this project, Pavel, and our mentor, Dr. Lubsky at Baylor. Thank you.